Another device to prevent self-pollination is the production of unisexual flowers. If both male and female flowers are present on the same plant, such as castor and maize monoecious, it prevents autogamy but not ketogamy. In several species such as papaya, male and female flowers are present on different plants. That is, each plant is either male or female, dioicy. This condition prevents from both autogamy. After completing a brief account of sexual reproduction of flowering plants, it would be worth attempting to comprehend the enormous reproductive capacity of some flowering plants by asking the following questions. How many eggs are present in an embryo sac? How many embryo sacs are present in an ovule? How many ovules are present in an ovary? Pollen-pistil interaction. Pollen does not guarantee the transfer of the right type of pollen. Compatible pollen of the same species as the stigma. Often, pollen of the wrong type, either from the other species or from the same plant, if it is self-incompatible, also land on the stigma. The pistil has the ability to recognize the pollen, whether it is of the right type, compatible, or of the wrong type, incompatible. If it is of the right type, the pistil accepts the pollen and promotes post-pollination events that leads to fertilization. If the pollen is of wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between pollen grain and the pistil. This dialogue is mediated by chemical components of the pollen interacting with those of the pistil. It is only in recent years that botanists have been able to identify some of the pollen and pistil components and the interaction leading to recognition, followed by acceptance or rejection. As mentioned earlier, following compatible pollination, the pollen grain germinates on the stigma to produce a pollen tube through one of the germ pores. The contents of the pollen grain move into the pollen tube. Pollen tube grows through the tissues of the stigma and style and reaches the ovary. You would recall that in some plants, pollen grains are shed at two-cell stage, a vegetative cell and a generative cell. In such plants, the generative cell divides and forms two male gametes during the growth of pollen tube in the stigma. In plants which shed pollen in the three-cell stage, pollen tubes carry the two male gametes from the beginning. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus. Many recent studies have shown that filiform apparatus present at the micropylar part of the synergids guides the entry of pollen tube. All these events, from pollen deposition on stigma until pollen tube enters the ovule, are together referred to as pollen-pistil interaction. As pointed out earlier, the pollen-pistil interaction is a dynamic process involving the pollen recognition, followed by promotion or inhibition of the pollen. The knowledge gained in this area would help the plant breeder in manipulating pollen-pistil interaction, even in incompatible pollinations, to get desired hybrids. You can easily study pollen germination by dusting some pollens from flowers such as pea, chickpea, crotalaria, balsam, and vinca on a glass slide containing a drop of sugar solution about 10%. After about 15 to 30 minutes, observe the slide under low power lens of the microscope. You are likely to see pollen tubes coming out from the pollen grains. A breeder is interested in crossing different species and often genera to combine desirable characters to produce commercially superior varieties. Artificial hybridization is one of the major approaches of crop improvement program. In such crossing experiments, it is important to make sure that only the desired pollen grains are used for pollen and the stigma is protected from contamination from unwanted pollens. This is achieved by emasculation and bagging techniques. If the female parent bears bisexual flowers, removal of anthers from the flower bud before the anther dehisces using a pair of forceps is necessary. This step is referred as emasculation. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. This process is called bagging. When the stigma of bagged flowers attain receptivity, mature pollen grains collected from anthers of the female parent are dusted on the stigma, and the flowers are rebagged and the fruits are allowed to develop. If the female parent produces unisex flowers, there is no need of emasculation. The female flower buds are bagged before the flower opens. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flower is rebagged. Double fertilization. After entering one of the synergids, the pollen tube releases the two male gametes into the cytoplasm of the synergid. One of the male gametes moves towards the egg cell and fuses with its nucleus, thus completing the syngamy. This results in formation of a diploid cell, the zygote. The other male gamete moves towards the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus, PEN. As this involves the fusion of three haploid nuclei, it is termed triple fusion. Since two types of fusion, syngamy and triple fusion, takes place in the embryo sac, the phenomenon is termed as double fertilization, and the event is unique to flowering plants. The central cell after triple fusion becomes the primary endosperm cell, or PEC, and develops into the endosperm, while the zygote develops into the embryo. Post-fertilization structures and events. Following double fertilization, events of endosperm and embryo development, maturation of ovules into seeds and ovary into fruit are collectively termed post-fertilization events. Endosperm. Endosperm development precedes embryo development. Why? The primary endosperm cell divides repeatedly and forms triploid endosperm tissue. The cells of this tissue are filled with reserve food materials and are used for the nutrition of the developing embryo. In the most common type of endosperm development, the PEN undergoes successive nuclear divisions to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is a cellular endosperm. Endosperms may either be completely consumed by developing embryo, example pea, groundnut, beans, before the seed maturation, or it may persist in mature seeds, example castor and coconut, and be used up during seed germination. Split open some seeds of castor, peas, beans, groundnut, fruit, or coconut and look for the endosperm in each case. Find out whether the endosperm is persistent in cereals, wheat, rice, and maize. Embryo. Embryo develops at the micropylar end of the embryo sac, where the zygote is situated. Most zygotes divide only after a certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to developing embryo. Though the seeds differ greatly, the early stages of embryo development or embryogeny are similar in both monocotyledons and dicotyledons. Figure 1.13 depicts the stages of embryogeny in a dicotyledonous embryo. The zygote gives rise to the pro-embryo and subsequently to the globular, heart-shaped and mature embryo. A typical dicotyledonous embryo, figure 1.14a, consists of an embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledons is the epicotyle, which terminates with the plumule or stem tip. The cylindrical portion below the level of the cotyledons is hypocotyle, that terminates at its lower end in the radicle or root tip. The root tip is covered with a root cap. Embryos of monocotyledons possess only one cotyledon. In the grass family, the cotyledon is called scrutellum, that is situated towards one side lateral of the embryonal axis. At its lower end, the embryonal axis has the radicle and root cap enclosed in an undifferentiated sheath called coleorhiza. The portion of the embryonal axis above the level of attachment of scrutellum is the epicotyle. Epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure, the coleoptile. Soak a few seeds in water, say of wheat, maize, peas, chickpeas, groundnut, overnight. Then split the seeds and observe various parts of the embryo and the seeds.
Seed. In angiosperms, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. Seeds are formed inside fruits. A seed typically consists of seed coat, cotyledon, and an embryo axis. The cotyledons of the embryo are simple structures, generally thick and swollen due to storage of food reserves, as in legumes. Mature seeds may be non-albuminous or ex-albuminous. Non-albuminous seeds have no residual endosperm, as it is completely consumed during embryo development. Example, pea, groundnut. Albuminous seeds retain a part of the endosperm as it is not completely used up during embryo development. Example, wheat, maize, barley, castor. Occasionally, in some seeds, such as black pepper and beet, remnants of nucellus are also persistent. This residual persistent nucellus is the perisperm. Integuments of ovules harden as tough protective seed coats. The microphyle remains as a small pore in the seed coat. This facilitates the entry of oxygen and water into the seed during germination. As the seed matures, its water content is reduced and seeds become relatively dry, 10 to 15% moisture by mass. The general metabolic activity of the embryo slows down. The embryo may enter a state of inactivity called dormancy, or if favorable conditions are available, adequate moisture, oxygen, and suitable temperature, they germinate. As ovules mature into seeds, the ovary develops into a fruit. That is, the transformation of ovules into seeds and ovary into fruits proceeds simultaneously. The wall of the ovary develops into the wall of fruit called pericarp. The fruits may be fleshy as in guava, orange, mango, etc., or may be dry as in groundnut and mustard, etc. Many fruits have evolved mechanisms for dispersal of seeds. Recall the classification of fruits and their dispersal mechanisms that you have studied in earlier classes. Is there any relationship between number of ovules in an ovary and the number of seeds present in a fruit? In most plants, by the time the fruit develops from the ovary, other floral parts degenerate and fall off. However, in a few species such as apple, strawberry, cashew, etc., the thalamus also contributes to fruit formation. Such fruits are called false fruits. Most fruits, however, develop only from the ovary and are called true fruits. Although in most of the species, fruits are the results of fertilization, there are a few species in which fruits develop without fertilization. Such fruits are called parthenocarpic fruits.
Banana is one such example. Parthenocarpy can be induced through the application of growth hormones, and such fruits are seedless. Seeds offer several advantages to angiosperms. Firstly, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seeds have better adaptive strategies for dispersal to new habitats and help the species to colonize in other areas. As they have sufficient food reserves, young seedlings are nourished until They are capable of photosynthesis on their own. The hard seed coat provides protection to the young embryo. Being products of sexual reproduction, they generate new genetic combinations leading to variations. Seed is the basis of our agriculture.
Dehydration and dormancy of mature seeds are crucial for storage of seeds, which can be used as food throughout the year and also to raise crop in the next season. Can you imagine agriculture in the absence of seeds or in the presence of seeds which germinate straight away soon after formation and cannot be stored?
How long do the seeds remain alive after they are dispersed? This period again varies greatly. In a few species, the seeds lose viability within a few months. Seeds of a large number of species live for several years. Some seeds can remain alive for hundreds of years. There are several records of very old yet viable seeds. The oldest is that of lupin, Lupinus arcticus, excavated from the Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. A recent record of 2,000 years old viable seed is of the date palm, Phoenix dactylifera, discovered during the archaeological excavation at King Herod's Place, near the Dead Sea. After completing a brief account of sexual reproduction of flowering plants, it would be worth attempting to comprehend the enormous reproductive capacity of some flowering plants by asking the following questions. How many eggs are present in an embryo sac? How many embryo sacs are present in an ovule? How many ovules are present in an ovary? How many ovaries are present in a typical flower? How many flowers are present on a tree? And so on. Can you think of some plants in which fruits contain very large number of seeds? Orchid fruits are one such category, and each fruit contains thousands of tiny seeds. Similar is the case in fruits of some parasitic species such as Orobanche and Striga. Have you seen a tiny seed of ficus? How large is a tree of ficus developed from that tiny seed? How many billions of seeds does each ficus tree produce? Can you imagine any other example in which such a tiny structure can produce such a large biomass over the years? Apomixis and polyembryony. Although seeds in general are the products of fertilization, a few flowering plants such as some species of asteraceae and grasses have evolved a special mechanism to produce seeds without fertilization called apomixis. What is fruit production without fertilization called? Thus, apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reduction division and develops into the embryo without fertilization. More often, as in many citrus and mango varieties, some of the nuclear cells surrounding the embryo sac start dividing, protrude into the embryo sac and develop into embryos. In such species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is referred to as polyembryony. Take out some seeds of orange and squeeze them. Observe the many embryos of different sizes and shapes from each seed. Count the number of embryos in each seed. What would be the genetic nature of apomictic embryos? Can they be called clones? Hybrid varieties of several of our food and vegetable crops are being extensively cultivated. Cultivation of hybrids has tremendously increased productivity. One of the problems of hybrids is that hybrid seeds have to be produced every year. If the seeds collected from hybrids are sown, the plants in the progeny will segregate and do not maintain hybrid characters. Production of hybrid seeds is costly, and hence the cost of hybrid seeds becomes too expensive for the farmers. If these hybrids are made into apomicts, there is no segregation of characters in the hybrid progeny. Then the farmers can keep on using the hybrid seeds to raise new crop year after year, and he does not have to buy hybrid seeds every year. Because of the importance of apomixis in hybrid seed industry, active research is going on in many laboratories around the world to understand the genetics of apomixis and to transfer apomictic genes into hybrid varieties.